Welcome viewers. Today we will start discussing about classification of viruses. Before starting the classification, let's peep into the microbial world. Microbial world comprises of living organisms as well as non-living particles called viruses, viroids and prions. What are viruses? Viruses are obligate intracellular parasites. They comprises of nucleic acid, and this nucleic acid may be DNA or RNA in nature. This nucleic acid is covered up by a protein coat called capsid. Complex of nucleic acid with the protein coat is called a nucleocapsid. Some viruses are naked viruses, they don't have any extra envelope. But there are certain enveloped viruses wherein we will see then they have in additional an envelope made up of lipids. This lipids envelope is mainly made up of host cells. Now let's start why the study of viruses is so important. There are many human diseases which are caused by viruses. The most popular one is called SARS, which has created trouble worldwide. Then we have seen the epidemic of H1N1 influenza virus in 2009. All of us are very much aware of AIDS, which is again caused by a virus called retrovirus. This is the disease part. Apart from this, viruses are playing many more vital roles. For example, whenever they infect any bacterial cells, these viruses are called bacteriophages. These bacteriophages are actually responsible for transfer of genetic material from one bacteria to other bacteria. Hence, they can contribute immensely to the evolution of bacteria. Next, we have seen that viruses are used to study molecular biological aspects like studying the mechanism of transcription, translation and DNA replication. We use viruses to understand the molecular tidbits of these mechanisms. Apart from this, there are various viral enzymes which are used every now and then for carrying out recombinant DNA technological experiments. We have seen that viruses interact with cellular microbes and contribute to the movement of organic matter from particulate forms to dissolved forms. Now, let's look into the minute properties of virus. Before that, we will go to the history of viruses, wherein we will see the first discovery was done by Ivanowsky in the year 1892. He was working on tobacco mosaic virus, and he could find that if the leaves of the diseased plant were extracted and this extract was used to infect the fresh plant, then the fresh plants were also getting infected. He purified the extract by using Chamberlain filter. But even though that filtrate was able to cause the infection, he found that this agent is surely not bacteria, because the membrane that he was using to filter the extract was capable of separating the bacteria. Therefrom, he could infer that the causative agent is surely not a bacteria. But he couldn't comment anything about the nature of the pathogen. He simply thought that the cause was a toxin. Later on, another scientist who is well known as the father of virology is Bacteriology, who worked independently, and he could establish that this agent, which was smaller than bacteria, which could not be filtered through the membranes, is a virus. he called it a virus contagium vivium fluidum. And finally, he was the person who coined the term virus. Next important discoveries was done by Loeffler and Froch. What he did, he found that foot-mouth disease of cattle was caused by viruses. Later on, many scientists contributed immensely, and they could find that viruses are behind causes of various cancers and many diseases. Finally, we could see that Twort in 1915 first time speculated that bacteria can also be attacked by viruses. This thing was further justified by Felix Deherel, who established the existence of bacterial viruses. That really changed the story of science, because people were fighting very hard to combat bacteria, and they were designing multiple varieties of drugs to combat bacteria. And now they have got an agent which can infect the bacteria and can kill the bacteria. So they were very much anxious to learn and to know more and more about this type of viruses that infect bacteria, or which we are called as bacteriophages. Then we could also acknowledge the contribution by Stanley in 1935, wherein he could first time crystallize a virus, and the first crystallized virus was tobacco mosaic virus. Crystallization of the virus actually helped us to look into the structure of the virus. Therefrom, we could infer that the virus is actually a complex of nucleic acid and proteins. So this thing was finally proved by Norman Peary when he separated tobacco mosaic virus into proteins and nucleic acids. This is a brief journey of the development of virology. The work is still in progress. So let's look into the important properties of a virus that makes it different from the living world. So viruses are non-living entities, we all know. They are not organisms, number one. Number two, they are just bits of nucleic acids surrounded by a protein coat. Then we have seen that they don't have any ribosomes, as other living organisms do have ribosomes. It means they don't have their own protein biosynthesis machinery. They are depending on the host cells for getting the supply of these things. Next, they cannot replicate outside the host cells. They can replicate only inside the host cells. Next, these agents can infect all forms of life, starting from bacteria, plants, animals. All of them are susceptible to infection by viruses. Now, the number of viruses were increasing day by day. It has almost crossed the count of 5,000. This was the high time when people started thinking that we need to classify the viruses and we need to cluster them into different groups. Then virologists were looking into the other classification systems which were available for the classification of the cellular organisms in the microbial world. Let's have a look into the classification systems and what is the need for a classification system to be present. First, the classifications of any living organisms are done to bring some order, means to bring some sort of organization. An organization wherein the organisms of similar criteria or having similar characteristics can be clustered together into one group. The second importance of classification is to be able to communicate with each other so that the members of different group can communicate among each other. And the third important thing is to assemble like members with each other. So this was the requirement for any classification system. If we look into the classification system that were existing in the olden days, the first name that comes in our mind is R.H. Whittaker. His famous five kingdom classification that was launched in 1969 comprises of Plantae, Animalia, Fungi, Protista and Prokaryote. But this classification system faced certain drawbacks, namely, it doesn't reflect the recent genetic insights of the ribosomal RNA data. And number two, plants and animals were more closely related to each other than archaea are to the bacteria. To overcome these deficits or these deficiencies, in the year 1977, Carl Woese came with three domain classification system, wherein he said that the three domains of life are bacteria, archaea and eukarya. Unfortunately, even while launching this new classification system, viruses couldn't find any place into this classification system. So, it was really very much necessary to look for a separate classification system for viruses. As they were not living entities, they couldn't find any place in the existing classification systems. So prior to the invention of sophisticated instruments, when viruses were unable to be seen by the scientists, the only characteristic that was known about viruses was their ability to cause infections. So as they were considered as agents that can cause infections, so they started classifying the viruses on the basis of the diseases that the viruses can cause. So in this way we can broadly categorize them into four groups. Number one is called enteric viruses, or viruses that cause enteric disorders or which replicate inside the enteric system of the human beings. Second class of viruses were those that causes respiratory disorders. They were called respiratory viruses. Third was called zoonotic viruses. This was a typical type of virus which mainly infects other animals, but during their course of replications, sometimes they may infect human beings, such as rabies. And the fourth category was sexually transmitted viruses like herpes virus, papilloma virus, and HIV. Though this classification appeared to be a good one, as they were taking into account the mode of infection of viruses onto different hosts. But the problem lies that there were innumerable viruses that were not at all responsible for causing any disease. And sometimes we could also find that there are certain viruses that can infect different varieties of hosts. And in one host, they might be causing one disease, and in other host, they are causing other diseases. So, this type of classification was really not very helpful. On the other hand, we were not able to identify all types of viruses that are responsible for causing diseases. For example, we were mainly focusing on the human system or on those viruses that are causing disease to the domestic animals. So in this classification, we could find that many viruses couldn't find their place. So they thought of looking for some other sort of classification. So the next classification was launched by Holmes in the year 1948, where he classified the viruses on the basis of the host organisms. So he broadly classified them into three groups under the order of virials, where he called group 1 viruses as phaginae, that is viruses that attack bacteria. Second one was phytophaginae, viruses that attacks plants. And the third one was zoophaginae, that is viruses that attacks animals. This was a quite sensible type of classification, but still it has got certain drawbacks. Namely, some viruses are restricted in their host range, infecting only one species. So, this type of viruses can be nicely classified under the classification scheme. But there are some viruses whose host ranges were very broad. For example, there are certain viruses which can infect both plants and insects. Then the question arises, then how I am going to place that particular virus into which group? So this was a serious drawback, and studying a single virus which infects a single species tells nothing about the fundamental nature of the potentially many other viruses which also infect the same host. It means there might be more than one varieties of viruses that are infecting the same host. But basically, if we look into the fundamental characteristics of the viruses, we couldn't find any similarity between them. So this type of classification was really not very worth it. Then people started thinking that we need to have some more modified type of classification wherein the clustering is quite sensible. Then came the development of electron microscopy, wherein we could see the viruses under the electron microscope. So seeing is believing, and this was the thing that classification on the basis of virus particle morphology was launched. So viruses were visualized in two different ways. Number one was that site of formation of the virus within the cell, as it was seen by thin sectioning techniques, because by that time the tools to cut the cells were already discovered. Then the second method was the symmetry property of the virus, as it was seen by negative staining. This classification of viruses on the basis of morphology, this classification really depends a lot on the contribution of Watson and Crick's X-ray diffraction studies. They did this X-ray diffraction studies in 1956. Their proposition says that viruses would be composed of a centrally placed nucleic acid fraction and an external protein coat or shell. The second proposition says that protein shell composed of small identical subunits of proteins arranged identically on its surface. Two types of geometric arrangements would occur. Identical subunits would be arranged in the form of a helix, or in other ways the protein subunits are arranged in cubic symmetry. These concepts actually helped in establishing the real structure of the viruses. Therefore, we could see that the unique structural features of viruses that were later seen under the electron microscope, they said that there are no organelles, no cytoplasm, no cell nucleus or no nucleoid. It comprises of basic component, a nucleic acid genome and a surrounding protein coat. The genome contains either DNA or RNA, but never both. Then protein coat of virus, which they called capsid, was made up of individual protein subunits that was called as capsomeres. And the complex of capsid with nucleic acid was called as nucleocapsid. Then many viruses were surrounded by flexible membranes called envelopes. So on the basis of existence of envelope or non-existence of envelope, they were grouped into two categories as enveloped viruses or non-enveloped viruses. And on the basis of their capsid symmetry, they were classified into three different groups called icosahedral capsids, helical capsids, or complex capsids, or binal capsids. So these were the three broad categories of classifying the viruses on the basis of their morphology. So let's start with icosahedral capsids. Icosahedral capsids actually comprises of two words. Icos means 20 and edros means side. It means that icosahedral capsid comprises of 20 sides, wherein we could see that 20 equilateral triangles are arranged to form a structure called icosahedron. It means within this structure there was maximum space for packaging of the viral genome. As you can see that virus genome is being packaged inside this capsid, and this viral capsid has got three different folds of axes. One was 3-fold axis of symmetry, 5-fold axis of symmetry, and 2-fold axis of symmetry. Example of this variety of icosahedral capsid bearing viruses is adenovirus. Next, we come to the viruses with helical capsids. The most important example of helical capsid virus is tobacco mosaic virus, wherein we could see that their genome is packaged in a helical fashion, and these small units of the capsid we are calling as capsomeres. The third and the most important variety of virus classification on the basis of morphology was the complex viruses or binal viruses. These viruses were mostly bacterial viruses, or the viruses that infect bacteria or bacteriophages. The most important example is T4 virus, wherein we could see that it comprises of a icosahedral head and a helical tail, means two structures, the icosahedral structure as well as helical structure. Both of them are present in case of bacteriophage T4. Here we could see that it has got a protein coat. Within the protein coat, the viral genome is being packaged and the tails are there, and this tail is mainly made up of helical morphology. So this type of classification was much more sensible as compared to the previous classification systems, because herein at least we were clustering the viruses on the basis of their morphologies. But still there are many drawbacks, that knowing the shape of a virus particle does not allow us to predict anything about its biology, its pathology, or its molecular biology of similarly shaped viruses. It's not necessary that if the viruses look alike, then if their morphologies are same, we can't say that their mode of infection or their genetic material or their ability to cause disease or their ability to promote any event is the same. So that's why this classification is not still scientifically perfect. Moreover, two viruses with very similar morphologies may differ in all of their fundamental characteristics. We could see under the electron microscope that the viruses are morphologically identical, but when we are working with them, we could find that their host ranges are different, that their mode of replication, their mode of transcription, their mode of executing their metabolic pathways within the host cells are drastically different. So this type of classification is still not a perfect classification. But even though it faces certain drawbacks, for a preliminary basis of classifying a virus, this classification still stands correct, that whenever we start working on a new virus, the first thing that we do with it, that we go for an electron microscopic study of the virus and look into the structural criteria and classify it into one of these three groups. So, to start up the study of any new virus, this classification of the virus on the basis of morphology still stands true. But we need to look for other classification systems that depends on the nucleic acid content or that depends on certain characteristics that can regulate the execution of important events carried on by the virus during its life cycle inside their suitable hosts.